Good morning. It's September 26th, and this is your daily brief in science. Here's everything you need to know. The tragic tale of the Franklin expedition, which set sail in 1845 in search of a navigable Northwest Passage, has taken a new turn with the recent identification of Captain James Fitzjames's remains. All 129 crew members are presumed dead after their ships, HMS Erebus and HMS Terror, became trapped in ice, leading to a desperate struggle for survival. Fitzjames served as second-in-command after Sir John Franklin's death, and recent research has successfully matched a tooth found on King William Island to a cheek swab from one of his descendants. This identification marks Fitzjames as the second of the 105 crew members to be positively identified, following engineer John Gregory's identification in 2021. Analysis of the remains revealed cut marks, indicating that survivors, including Fitzjames, resorted to cannibalism during their dire circumstances. Researchers noted that while survival cannibalism is often viewed negatively, it can be contextualized by the extreme desperation faced by the sailors. These findings shed new light on the tragic circumstances surrounding the expedition and underscore the importance of meticulous archaeological research. The research challenges sensationalized narratives about the expedition and highlights the significance of testimonies from indigenous Inuit people who reported seeing survivors. Fabienne Teteroux, a naval historian, played a crucial role in identifying Fitzjames's remains, while Doug Stenton, an archaeologist involved in the study, emphasized the need to understand the sailors' plight in the context of their actions. Efforts to find descendants of Franklin expedition members have led to the discovery of Fitzjames's relatives, who expressed excitement upon learning about the identification. The wrecks of HMS Erebus and HMS Terror were discovered in 2014 and 2016, respectively, providing crucial context for understanding the fate of the expedition. A recent review published in the journal Addiction has brought to light important findings regarding buprenorphine treatment for opioid use disorder. The study revealed that the rate of buprenorphine-precipitated withdrawal in adults is low, ranging from 0 to 13.2%. This is particularly significant given that nearly 70% of the 108,000 overdose deaths in the U.S. in 2022 were linked to fentanyl, a substance that is notably more potent than heroin. Dr. Nora D. Volkow from the National Institute on Drug Abuse emphasized the necessity of adapting opioid treatment strategies to meet the challenges posed by the ongoing fentanyl crisis. The study indicated that adults on higher daily doses of buprenorphine, specifically those exceeding 16 to 24 milligrams, experienced a 20% longer time before requiring emergency care compared to those on lower doses. However, access to these higher doses remains limited due to various barriers, including restrictive state laws and insurance policies. Despite the favorable evidence supporting higher doses of buprenorphine, stigma surrounding opioid treatment and prevailing medical culture contribute to its underprescription. Future research is crucial to understand the effects of high-dose buprenorphine across diverse populations and to evaluate long-term treatment retention and health outcomes. The analysis reviewed insurance claims data from over 35,000 individuals diagnosed with opioid use disorder who began buprenorphine treatment between 2016 and 2021. Lead author Dr. Caroline Gregory from the University of Ottawa noted that the fear of precipitated withdrawal should not deter the use of buprenorphine, as it remains an evidence-based treatment. There is optimism that the growing body of evidence will prompt the FDA to revise its dosing recommendations, which currently influence insurance practices. Preventing the need for urgent health care can significantly benefit the health and recovery of individuals with opioid use disorder, as highlighted by Dr. Bradley D. Stein. A recent study published in the journal Nature has unveiled a more intricate evolutionary history of jaw structures in mammals. This research indicates that the extinct cynodont species Riograndia guibensis had a mammalian-style jaw joint 17 million years earlier than the previously oldest known example. In contrast, another cynodont, Brachyllodon quadrangularis, did not exhibit this advanced jaw feature. Lead author James Rawson pointed out that the evolution of the mammalian jaw joint marks a critical moment in mammalian evolution. Researchers employed CT scanning technology to reconstruct jaw joint anatomy, revealing that Rio Grande's jaw structure closely resembles that of modern mammals, despite Brasilodon being more closely related to living mammals. The study also re-examined Brasilodon's jaw joint morphology, which retains the ancestral quadrate articular joint and lacks the dentary squamosal articulation found in mammals. This unique jaw hinge, formed by the dentary and squamosal bones, allows mammals to chew more precisely than other vertebrates. Dr. Agustin Martinelli emphasized the importance of Brazilian fossils in enhancing our understanding of mammalian evolution, with ongoing multinational collaborations expected to yield further insights. Professor Marina Soares noted the unique diversity of cynodont forms in Brazil, which are vital for studying early mammalian evolution. The two cynodont species in question lived during the late Triassic epoch, around 225 million years ago. These findings necessitate a re-evaluation of existing theories about the evolution of the mammalian jaw hinge. Jie-Shi Luo from the University of Chicago described this discovery as significant, potentially reshaping our understanding of mammalian jaw evolution, although gaps in anatomical interpretation persist due to limited fossil availability. A groundbreaking study from UCLA has uncovered how specific brain cells encode time and experiences, shedding light on the mechanisms behind memory formation and future predictions. This research involved a three-phase experiment where participants engaged in tasks related to images displayed on a pyramid-shaped graph. Over the course of 40 minutes, participants recognized approximately 120 images, allowing researchers to observe patterns of neuronal activity. During the experiment, researchers noted how neurons in the hippocampus and entorhinal cortex aligned their activity with the sequencing of the presented images. The study revealed that these neurons adjusted their firing patterns based on the order of images displayed, indicating a progressive representation of experiences. Notably, the findings suggest that these neurons can anticipate subsequent images, highlighting the brain's ability to predict future events based on learned patterns. The researchers found that specific brain cells fire in distinct patterns that reflect the order of a person's experiences, with these unique firing patterns persisting even after the experiences have concluded. They also proposed that neuronal replay mechanisms may play a role in encoding temporal structure, suggesting a time-compressed reactivation of sequences during breaks in tasks. Dr. Itzhak Fried emphasized the importance of recognizing patterns over time for effective memory formation and behavioral guidance. The implications of this study could extend to clinical applications, such as enhancing memory therapies by targeting specific neuronal patterns associated with significant memories. The lead author, Pavel Tasakowski, along with co-authors Guldamla Kalendar and Davide Ciliberti, published their findings in the journal Nature. Dr. Fried suggests these insights could inform the development of neuroprosthetic devices aimed at enhancing memory and cognitive functions. A promising breakthrough in cancer treatment is emerging from the University of Wollongong, where PhD student Alice O'Keefe is at the forefront of developing innovative magnetic nanoparticles aimed at targeting cancer cells. These nanoparticles, known as Phe304/RGO nanocomposites, hold potential for a range of applications beyond cancer treatment, including energy storage, magnetic fluids, and catalysis. The synthesis of these nanoparticles involves a sophisticated process that combines the in situ formation of Phe304 nanoparticles with the reduction of graphene oxide into reduced graphene oxide using controlled microwave irradiation. This method is crucial for creating superparamagnetic iron oxide nanoparticles, or SPIONs, which are integral to the development of these composites intended for magnetic hyperthermia applications. Magnetic hyperthermia, or MHT, has been under investigation for over 3 decades as a potential treatment for glioma tumors. This technique uses magnetic nanoparticles to generate heat that can induce cancer cell death, presenting a novel approach to combating aggressive tumors like glioblastoma multiforme, or GBM, which is known for its poor prognosis and high resistance to conventional therapies. The nanoparticles are engineered to demagnetize and stop generating heat once they reach a specific temperature, referred to as the Curie point. This temperature is lethal to cancer cells while remaining safe for surrounding healthy tissue. When subjected to a magnetic field, These nanoparticles can produce sufficient heat to kill cancer cells at temperatures ranging from 42 to 47 degrees Celsius. To ensure safety and efficacy, the biocompatibility of these nanocomposites has been enhanced by incorporating polyethylene glycol, or PEG, which has shown low cytotoxicity on healthy cells in incubation studies. Various characterization techniques, including TEM, SEM, XRD, and XPS, have been employed to thoroughly analyze the structural and magnetic properties of the synthesized nanoparticles. The research not only highlights the potential of magnetic hyperthermia, but also discusses the challenges it faces and the future prospects for its application in treating GBM. Improvements in this area could significantly enhance the effectiveness of this innovative treatment strategy. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.